Back. In this section, let's learn a little bit about hemostasis. So hemostasis just means that we're going to stop the bleeding by forming a blood clot. And there's going to be three distinct steps for this. The first is known as vascular spasm or vasoconstriction. The second is the formation of a platelet plug. Now that's not a blood clot. This entire process will give us a blood clot. So the second step is a platelet plug made of only platelets. And the third step is the activation of coagulation cascade. So this entire process of coagulation or blood clotting, which results in hemostasis, normally takes about 5 to 6 minutes. So when you get a nosebleed and you hold your nose for 1 minute or 2 minutes and then you let go, do not be surprised if your nose starts bleeding again because you're normal. You take 5 to 6 minutes to finish your blood clotting. So refresh your memory. We're going to go through 3 steps in this order. The first step being the vasoconstriction, where the injured blood vessel is going to narrow, thereby slowing the blood flow. Just think, an accident on the 405 where instead of being 5 lanes wide, you're down to 2 or 3 lanes. The second step being the formation of a platelet plug. And I need you to realize this only happens at the site where the blood vessel itself is injured, because that's what's triggering this. And then the third step, that coagulation cascade, is going to occur, the end result of which we are going to have this protein known as fibrin, which forms this mesh, which then when the blood is traveling by it will trap the other blood cells. So as you can see in this picture, the aqua-colored structures are the fibrin fibers. The purple are the platelets in aggregates because they have been activated when the platelet plug was formed. And then you can see red cells, which are red blood cells, and then of course white blood cells would get caught as well. So let's get started. Step 1, vasospasm. When a blood vessel is injured, it's going to cause release of chemicals locally. And one of these chemicals is going to cause vasoconstriction. Now realize this only lasts at most a few minutes. But the net result of this is going to start the processes leading us to step 2 and step 3. Now, this is a neurogenic reflex that is also mediated by pain receptors, so you can get a more profound vasoconstriction if pain receptors are damaged. And so when we're looking at what kind of chemicals might be involved at the site of injury, when platelets are going to be involved, they are going to release 2 different chemicals, thromboxane A2 as well as serotonin. So the serotonin is going to cause some vasoconstriction. And the thromboxane A2, if we go back to our arachidonic acid pictures, remember on one method using those COX enzymes was to form prostaglandins, and one of the 3 types was thromboxane A2, which directly is in platelets and can cause multiple things. Increase in platelet IP3, so we have a second messenger, which we're not going to talk the details of that. It's going to give us platelet aggregation. We will talk about that without talking about chemical reactions that are happening, and also the vasoconstriction from the serotonin being released as well. So that leads us to step 2, platelet activation. So normally the inside of a blood vessel wall is very smooth and it has a coating, kind of like on the outside of a brand new car. You've got this coating which keeps all the bad stuff from getting into the paint. Well, it's got this coating on the inside of the blood vessel wall, which essentially is a platelet repellent, known as prostacyclin, but you don't need to know that word, okay? And so this platelet repellent just kind of keeps the platelet flows moving past them in a smooth method. Now, of course, when the blood vessel wall is damaged, that portion will not have this platelet repellent on it. And what actually happens at the site of damage below those epithelial cells, that damaged blood vessel wall is exposing what is underneath the epithelium. And one thing that is underneath them are collagen fibers. And so when the platelet comes cruising along, and because it doesn't have that Teflon skidding to go past it, it cruises along at the site of injury where the collagen is, and that causes that platelet to change its shape once it hits a collagenous fiber. And so this shape has got all these little points poking out all over the place, and that causes a whole bunch of reactions, one of which is going to be releasing several products, that serotonin and that thromboxane A2. And the net result is this platelet actually is going to become sticky on its surface. So it's going to be sticking locally where it hit the collagen. And it can move only now, not by the flow of blood, but by doing this amoeboid movement, where one of these little prongs kind of moves and it can pull the rest of itself behind it, just like an amoeba does. So this is going to lead us to forming what is known as a platelet plug. So when platelets are activated, they're going to release their serotonin and thromboxane A2, and the releasing of these chemicals is making the blood flow even slower in the area, as well as making the platelets stickier. And when a platelet comes and touches an activated platelet that's already changed shape, then that second platelet is going to change shape as well. And so we're going to get this positive feedback where we're going to have more and more platelet activations. And so what we're going to find is all these platelets adhering at the site of injury. Because remember, what triggered it was that subendothelial collagen, and those first platelets were stuck to it, and then the next platelet gets stuck to that one, and the next platelet gets stuck to that one. And so the platelet plug forms at the site of injury. And here's a picture of what a platelet plug kind of looks like. And you can see a lot of these platelets are kind of funky looking. So that is step 2. Step 3 is now coagulation cascade occurs. And so what this is a group of slightly more than a dozen proteins that float around in your plasma in an inactive state. And yes, we need a stimulus to start this. And so there are 2 different ways to start this pathway. The first is what's called the intrinsic pathway. And the intrinsic pathway is what occurs when you have damage to the blood vessel wall, and now the subendothelial collagen is exposed to the plasma, and that by itself can trigger this. And so once the first protein gets activated by the subendothelial collagen, then it can trigger the next one in the sequence, which will trigger the next one, and on and on and on. Now, there is a second pathway known as the extrinsic pathway, which we're not going to explore in this class. But if you continue on and take pathophysiology classes later on, you will learn about this. And basically, there's something in tissue called tissue factor. So even if you don't have subendothelial collagen, you can have release of tissue factor, which causes the extrinsic pathway to be activated. And so we will have proteins being activated, which then activate the next one on both the intrinsic and extrinsic pathways until they join, and they are both activating factor 10, which is the beginning of the final common pathway. So I don't want you to learn these factors, their names, any of their cofactors, like calcium, any of the enzymes, any of that kind of stuff. Once I get to something I specifically named, that's where you need to learn this. And so we are there at this point. In the final common pathway, what is going to happen is an inactive protein known as prothrombin will be activated to form thrombin, which is also known as factor 2, but you don't have to know that. But thrombin then activates fibrinogen, this big long protein, chops a piece off of it, and you are left with this protein known as fibrin, which is the very last thing that is formed in coagulation cascade. And with other factors, then what happens is these little pieces of fibrin get cross-linked among themselves, and we are going to have the finishing parts of this coagulation cascade. So as you can see, if we've got 13 different things that need to be activated all in a series, this is going to take some time. And that's why it is normal for blood clotting to take 5 to 6 minutes total. So this final common pathway is the important part, where thrombin is going to activate fibrinogen to fibrin to form this fibrin clot, which cannot be dissolved in water. Okay, so prothrombin becomes thrombin, which then makes fibrinogen become fibrin, and now we have this cross-linked fibrin clot, where all these fibrin monomers are now stuck to each other, and this forms this meshwork, which is the framework for a blood clot, which you can see on this picture. So I ask you to stop this picture and look at this and talk yourselves through the entire process that we have talked about so far. Okay, continuing on, we're almost done with this section. I want to point out a couple more things. The first thing is written here in green. What you will notice that there is amplification that goes on. In other words, there are some clotting factors which, instead of like one of these forms one of these, then when this one becomes activated, it forms many of these. And so all these green arrows are showing all these sites where amplification occurs. So just by having collagen activating one molecule on the intrinsic pathway, we can form a large amount of fibrin. And so in addition to that, if we look right before fibrin, where we had our thrombin over here, which I said was really important, it's the thing that gives us our fibrin. So definitely learn thrombin, fibrinogen to fibrin. But something else that thrombin does is thrombin has positive feedback on the intrinsic factor itself, thereby activating it even more without more exposure or more damage. So all of this is greatly promoting a blood clot in the area. And so as you can see, this is all positive feedback, which eventually, if it was unchecked, would lead to your entire circulatory system forming one huge blood clot, and that would kill you. So come on back and we'll find out what happens, and I'll see you soon. Thank you.